Hi, everyone. Welcome to our monthly Science of All Protein seminar. My name is Bianca Dada, and I'm the Scientific Partnerships Manager at the Good Food Institute. For those of you who are here for the first time, welcome to our community. The Good Food Institute is a nonprofit think tank 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based, fermentation, or cultivated meat technologies, check out our open access resources at gfi.org/science. With that, I'd like to introduce today's speaker, who's joining us from a houseboat, very exciting. Dr. Alan Kuhl is the Katherine Holman Johnson Director of Stanford Biox and the Walter B. Reinhold Professor in the School of Engineering, focused on mechanical engineering. Her area of expertise is living matter physics and the design of theoretical and computer models to simulate and predict the behavior of living systems to benefit human and planetary health. Her recent work focuses on automated model discovery and integrates mechanical testing, mathematical modeling, and custom-designed physics-based neural networks. Alan's lab applies these technologies to characterize the mechanical signature of plant-based and animal meats. She's published more than 200 peer-reviewed journal articles and edited 2 books. In addition to her many, many scientific accomplishments, Ellen is an all-American triathlete, a multiple Berlin, Boston, and New York marathon runner, and a 3-time Kona Ironman World Championship finisher. Super impressive. Today, Dr. Kuhl will share her work on evaluating the mechanical signature of plant-based meats. Dr. Kuhl, thanks for being with us today. I'll turn the floor over to you.
Thank you, Bianca. Thanks for the kind introduction and thanks for the invitation. I'm very excited to be here and share with you our work. Bianca, gave you a nice introduction of what we do. The purpose for me, the main purpose is to get access to collaborators in this field. And the Good Foods Institute has been really amazing to connect us to people. And I hope that the seminar will continue connecting us to people in the field. We've only been in the space for about 2 years, and I'm going to share what we have done before and how we've gotten involved in trying to understand alternative protein products and also share our latest results. So thank you very much. I'm very excited. And I would like to start by just giving you a brief overview of what we do in the lab and what we've been doing for the good part of, say, 20 years or so. So our lab tests of materials and discovers models to predict the behavior of living systems. And we have initially done this for hard tissues like bone, and then gradually moved softer and softer. So we started with lung, and then the heart, and then skin, and most recently also the brain. And this has been very helpful because the brain is the softest of all tissues, and its stiffness is about the order of, think of silken tofu or so, or pudding. It's really, really soft. And it's challenging to test, and I'll get back to that in just a bit. And we've also modeled muscle quite a bit, and that's interesting because it's pretty similar to, say, a steak or so, because it has this fibrous microstructure. So all of this comes in handy, actually, for us to understand and trying to study animal and plant-based meat products. This is what we do for research. And then for teaching, we actually try to integrate research and education and bring these two together. So we have 2 classes at Stanford that seek to do that. And the first one is Mechanics of Biological Tissues by my colleague, Mark Levinson. And then my class, as Bianca already said, builds on this, and it's called Automated Model Discovery. That's a new class that we introduced 2 years ago. And you can see in the picture what we do. So we attempt to study all kinds of soft materials. And you can see 3 test devices here. So that's a tension, a pulling test device. And then many of you might be familiar with the rheometer for compression and shear testing. And then there's also a biaxial tester for flat, thin, 2-dimensional sheets. So the idea is if we can leverage students and classes, we can train more people in this field, and we can crowdsource a bigger community and draw on, actually, we have very amazing students, and we can draw on these students. They're all very excited, especially about plant-based protein. So in these classes, students do projects. And about 3 years ago, we started our research with a project in the biomaterials class. And these are 3 students who did this project. So this is Divya, Skylar, and Ethan. And since then, Skylar has actually moved on to become PhD student in my lab. And what I'm going to share is pretty much Skylar's PhD work over the last 3 years on plant-based meat. So what they did is they studied different meat products, and they were interested in trying to understand, can we use mechanical engineering to better understand meat? And to which extent does the the plant-based product mimic the features of animal meat? These are the kinds of data we collect, and I just wanted to give it to you, like show what it looks like. So these are tests in tension, compression, and shear, and these are 3 different products: Tofurky, chicken, and real chicken. And what we do is we push and pull and shear them by 10%. And you can see the data points here, and this is already massively cleaned up for anyone to use. So we get typically about a couple of thousand points out of one test, and we perform multiple tests. And when it says, for example, n equals 5 here, that means we've done 5 tests, and then we have averaged the results, and you could see here the mean out of these 5 tests, and that's what you can use to fit or to compare your product against or whatever you like. So we post all these data sets publicly on an open source platform. In this case, you can see here, it's a GitHub channel that's called Living Matter Lab Can. And if you go to that site, you will find all kinds of data sets like these. So this is what they look like. And our goal is to create an open source library that anybody can use, especially, say, small businesses who want to develop a product, and they don't know how it compares to other products. So we're very happy to help you test it if you have anything to test, and compare it against all the other products we've tested. If you want to test it yourself, you can also compare because all these data are available. And the idea that we have is we want to democratize discovery and innovation, and not only include big companies, big food, but also smaller startups that can then use all these data sets that we publicly share. I don't think this community needs motivation why we're interested in plant-based meat. In our lab, many people are actually vegan and strictly vegan, and the reasons that they cite are the energy conversion that it takes for animals or cattle to convert energy into edible meat, also the greenhouse gas emission and the sustainable impact, and then antimicrobial resistance that is increasing if we do only rely on animal agriculture. So our idea is we want to provide tools to understand how the mechanics of these products behave, because we think that in order to increase the number of people who switch their diet to more plant-based products, these plant-based products should have similar mechanics as animal meats. And so that's kind of the overarching idea. We started with these 8 products that you can see here. So we selected animal and plant-based products: hot dog, sausage, and turkey. And for comparison, we also used a firm and extra firm tofu product. So these are the 8 that we tested. So went to the supermarket, tested them. It's something students really like because they can get their hands on any kind of product by just going to the supermarket. Whereas when we test brains, it's really hard to get human brain samples. And so they have to sit in the lab and wait until something becomes available, and this is all readily available. And then they come up and say, how about we test gummy bears for comparison? So we could test all of that, and we did. And this is the first study I'd like to share. It's based on texture profile analysis, so a double compression test of the sample that's supposed to mimic the act of chewing twice. On the left, you can see these 8 products mounted in the device for double compression, and on the right, we also perform a torsion rheometer test. And on the top, you can see what the samples look like. So we have a biopsy punch that we punch through the sample, and then we extract a sample that's about 10 millimeters in height and 8 millimeters in diameter, and then we mount that into the device. In case you don't recall exactly what a texture profile analysis does, here you can see a quick overview. So on the top left, you can see an idealized texture profile analysis. It's a double compression test. In our case, we compress by 50%, so that's to half of the height. And then we release, we compress again and release. That's the idealized zigzag curve. In reality, it looks slightly different. So in the middle, you can see a real curve that we measured. It's a little more rounded, and also a little flatter at the bottom. But this is what you get when you really test and physically apply this. Out of these tests, typically people extract a number of features. I've listed the 6 that we look at here. For us as mechanical engineers, the first 2 are the most easily communicable to our community. So one is the stiffness. The stiffness is the slope of the stress strain curve, and it's measured in megapascal or kilopascal. The second is the hardness. That's the peak, the top of the first force and the first compression. And then there's a bunch of parameters that are just ratios of these areas, ratios of these times, and they don't have a physical unit. They just describe behavior beyond elasticity. So those are parameters that tell you how fast does the material break, how does it spring back when it's compressed, how resilient is it to failure. And the first two are really elastic parameters. So those are actually a lot more relevant to our community. So this is the result of the texture profile analysis for 6 parameters that we looked at. And it's actually for the 8 products. So every time you see an A here, it's an animal product. You see a P as a plant product. And then H is hot dog, S is sausage, T is turkey. And we have firm and extra firm tofu also. And what stands out is that plant turkey has on all charts the highest value. So the stiffness of plant turkey is on the order of 470 kilopascal, which is about 10 times stiffer than the softest product, which is firm tofu in this case. And interestingly, the animal products fall right within this range. So they are framed with the plant-based products from above and from below, and all animal products are in between. So that's actually telling us that we can mimic the stiffness of the animal products very well with plant-based products. This is the rheology, so it actually confirms what we've just seen. The turkey product, plant-based, is the stiffest, and the tofu products are the softest, and anything else is sandwiched in between, and this is confirmed just by a different type of test. And this is a summary of the hot dogs and the sausage product with the animal on the left and the plant-based product on the right. You can see a summary of all the features that we have extracted. And actually, you can see that the plant-based meat successfully replicates the elastic properties of processed animal meat. Now, there's one thing I should mention. This is processed meat. This is not a raw steak. So we have intentionally selected products that mimic the plant product. So a sausage is equally minced and it's equally processed as plant product. And what's interesting and important to realize is that they all have very long ingredient lists. So this is not plain ingredients. This has a lot of materials embedded. So there's a lot of stabilizers, there's a lot of gums, there's a lot of additives that try to make these products actually stiff. So you can tune the stiffness by adding those. And a lot of people are actually opposing plant-based products, as you probably know, because of these long ingredient lists. And they've been very controversially discussed because of their ultra-processed nature. So then we actually wanted to test it in tension, compression, and shear, like Bianca indicated in the beginning. And you can imagine that it's very difficult to test something in tension because you need to actually pull on it. And if something is very soft, like pudding, how would you even pull on a pudding? And so we designed a device. And the green thing you can see here is a holder that we 3D printed. And Skylar, Saint-Pierre, in our lab designed and printed it. And then we can glue the sample in these glass slides that you can see there. And then we can slide the glass slides into the holder and we can pull. And this allows us to perform actually high throughput tensile tests with these samples, as you can see here. So this is another test that we have added to this portfolio of tests to characterize these behaviors. Again, you can see the results here. So it's tension, compression, and shear. You have the 8 products. And again, just reiterating, if you're interested, you'll find the data in that link below. And if you don't get the time to write it down now, please reach out and we're happy to help you get started. This is what happens when you plot it. And to us, something stands really out. So these products are very reproducible. In the line, the solid line is actually the mean. And the shaded area is the standard error of the mean. And you can see it's very, very low. So many products have a very, very narrow tolerance range. So they're actually very, very reproducible in terms of their stiffnesses. So that's great. You can see the tensile test in the first row, the compression test in the second row, and the shear test in the third row. And what stands out is that the tensile and compressive stiffness is not the same. So these materials are asymmetric. They have a different behavior when you compress them than when you pull. So that's actually interesting. And that makes them very complex, but also from a material science point, very interesting to study. So if we move on from here, you can see the results in all one graph at the top: tension, compression, and shear. Again, all the plant products are in blue and the animal products are in red, sandwiched in between the two extremes, tofu and Tofurky. And then from that, we can do a linear regression and extract the stiffnesses. So that for material scientists is Young's modulus in tension, compression, and shear. And you can see that the dark blue has much larger bar than any of the other products, meaning that the plant turkey is really, really stiff, much stiffer than everything else, about 10 times as stiff as tofu. At the very last graph, K, you see something interesting. You can see that the ratio between tension and compression, and what you see is all animals products, so the red products, are above 1, meaning that they are stiffer in tension than they are in compression. And all plant products are reversed, so they are stiffer in compression than they are in tension, which is something we didn't expect and was quite surprising and an interesting material science type of result. So this is all what we would do in this very first class. And in the second class, we would discover models for the data. And what's important here is we don't just use a neural network. A lot of people, when they hear neural networks, they just think, okay, you throw data at a huge network and it gives you some fit of points. We don't want to do that. What we want to do is we want to discover physics in the data. And so we have designed a neural network that has physical features. And you can see some of them here, like symmetry and compressibility, consistency. And that gives us models that are interpretable and generalizable. And that's very important because we can now communicate a model that we find with anybody in material science, because chances are they have seen these models before for skin or for rubber or for wood or for any other structure that they need. So this is a cool feature in a way. And when we run this over the data, this is what we find. So we find one term that is very dominant. That's the dark brown. And that's the first invariant term. So that's a Neo-Hook linear model. So all these data have that term. And then on top of it, we find that tofu is very simple. It only has that one term. So you only see the dark brown on the very right. But all other materials have a second term. The ones on the very right have a light turquoise term, and that's called the Moni-Rivlin model. So that's a model that people have in the 1950s used for rubber, for example. And it's a very popular model. And then in between, for the animals, we find models that are more complex and that people haven't yet used in literature, combining brown and orange, brown and green, and brown and blue terms. So those are models that somehow are novel models that we have discovered. And we can go one step further and say, okay, what about if we find the very best model with only one term for each of these materials? Or what about the best model with only 2 terms? So we have this checkerboard plot where we plot any combination of 2 terms, and we use it to fit the data. And then we color code the little squares in this plot by the error of the fit. So a blue square means it's a very good fit of the model. A red means it's a very bad fit. And of course, we want a good fit. So if you look on the diagonal and you look at the bluest most square on each of the diagonals, you will find the model that best explains the data. And they are listed on the right. And they're also listed with their parameters. So you can actually use this now and predict the behavior of plant-based sausage in this range and even beyond. And then if you look at the entire graph, on the entire checkerboard, the dark most blue is the model with the best 2 terms combinations. So you can also see that. And actually, this is a nice way to illustrate this at first glance to find what's the best model with 1 term on the diagonal, what's the best model with 2 terms across the entire checkerboard, and how well do they perform. So summarizing everything I've just said, plant-based meat successfully replicate the elastic properties of animal meat. They can sandwich them in between the 2 extremes of plant turkey and tofu materials. But as a downside, they have a very long ingredient list, as you can see here, right? They use stabilizers, they use a lot of gum, they use just things to additives to tune these properties. So I'll show in just a bit how we can reduce this ingredient list with some materials. We then asked ourselves, well, what is this good for? Why do we need a model? And so we thought, how about, does this what we find mechanically relate to what we actually sense when we eat it? And so we conducted a texture survey and asked people to rank certain properties or features of these foods. And you can see the 8 of them here prepared from Tofurky on the stiffest on the left, to tofu on the right, in the middle, and then to all the animal products on the right. And so we surveyed 16 participants, and this seems to be a low number, but we are very lucky because we're at Stanford and we want to carry this test into the dining hall. So after the summer break, we're going to test all our students and have a number that would be probably in the hundreds, and have a much broader response. So this is a preliminary way to do this. And before we did this, we wanted to find out if this 16 people constitute a good study group. So we asked them if they're open to trying new foods and if they're attached to meat. So that's two surveys here in blue and in orange. And it turns out they're very open. I mean, they're students, so they are open to eating pretty much whatever comes their way. And they are moderately attached to meat. They're also very open to eat any plant-based alternative. And this is the survey, and this is the results. So let me walk you through this. What we did is we had 12 features, and we asked every person to eat a piece of a product and then say, this food is soft. On a scale from 1 to 5, do you think it's soft? Where 1 means no and 5 means very much. And we did this for all 12: soft, hard, and brittle, chewy, gummy, viscous, springy, sticky, fibrous, fatty, moist, and meaty. So there's a lot of products where adjectives or features where you can see a flat line with an S, and that means they're not significant. So there's no difference in these products. But interestingly, there's a huge difference in soft and hard, and that's actually what we're looking for. So if you want to taste the stiffness, then you would assume that softness and hardness vary in our perception of taste, and that's what they do. And very interestingly, remember the plant turkey in dark blue is the hardest when we eat it, and the two tofus in light blue are the softest when we eat it. So really this soft material is dominated by the tofus, and hardness is dominated by what also we found Tofurky to be the stiffest. That's in the first row, so that's the mechanics of it. And then there's another interesting finding in the bottom row, where people perceive the animal product as all three, the red bars, the most fatty. So all red bars are to the left of the fattiness study. And then the blue, the plant-based products are all to the right. And that's the same for meat-like, which is not surprising because they are actually meat. So they are scored the highest meat-likeness. So then we said, okay, how do these two correlate? How does what we taste correlate to what we measured before? And you can see in the left, the stiffness that we measured with our machine. And then in the middle and on the right, the stiffness that we tasted when we were eating it. And you can see if you compare the hardness to the stiffness, the order is almost identical with the same stiffest product, Tofurky, and the same softest product, tofu. And then if you look at the softness, it's just the same, but a flipped order because soft is the inverse of hard. And in other words, if you're a statistician, you can actually run correlation analysis and look at the Pearson correlation coefficient. It's 0.833, which is very close to 1. The p-value is 0.015, which means there's a very strong correlation between what we taste and what we measure. So that's great. So essentially, we can taste stiffness, and that would potentially allow us to replace sensory panels partly by mechanical tests. These tests are more reproducible, they're more reliable, they're very robust. Wherever in the world you perform them, they would give you the same results. Whereas if you have a tasting panel, it might depend on time of day, people who are tasting, age of the participants, location, all kinds of things, and it's not very reproducible. So this might be a good way to do it. And so we're trying to build a database on both that we are sharing so that people can use it. This was interesting because it was actually in the news, and the students, of course, like to be in the news. It was on ABC7 in November. And so here you can see the students of Skylar, Ethan, Jeremy, and Thibault together when we recorded that video, and then everybody in the lab trying to test and measure some of the products in the lab. We then, since November, continued to actually look at different type of products. So this is a study where we moved the block brick foods to two-dimensional deli slices. And you can see here again, we had 4 deli slices that were plant-based: plant turkey, plant ham, plant deli, and plant prosciutto, and compared them to 4 deli slices that were animal-based: animal turkey, chicken, ham, and prosciutto. And this is a study Skylar did with 3 undergraduates over the summer last year. And what's different in these products is you cannot test them in tension, compression, shear. because they're really like thin. You can't compress this little thin layer of food. So you would perform biaxial tests, and you can see this here. You hook them into the biaxial device, and you can see all 8 products helped. And you would pull on the hooks. So you'd only test them in the plane, but you can test different combinations of loading in the X and Y direction, which gives you a very broad picture of the behavior of this material. And then what's interesting out of this study is that, again, plant turkey, the same product now sliced, was the stiffest of all 8 products, similar to the first study. So it's actually confirming that even if we look at deli slices, we find similar and comparable results. This paper just got uploaded on arXiv, so you're very welcome to read it there. I could share some more results to this study, but really what I think is very interesting, might be interesting to you, is our very latest study that we have not yet published. So it's just raw study just coming out. We're going to submit the paper probably in 2 weeks. So this is very, very new results. And this is on fungi-based meat. I've talked about the ingredient list, and the advantage of really fungi-based meats, as you probably all know, is that they have very lean ingredient lists, and they're also naturally anisotropic. So you can see this here. There's a belief that these materials that are made of, or this meat that is made of mycelium, is naturally moist and fibrous, because mycelium is this fibrous root of the mushroom. And also that it's naturally meat-like, because mushrooms have this umami type taste. So we wanted to understand, is it actually true? So this is very, very recent. We just finished the last study. You can see what we did. We took a steak and we punched through it in the cross-plane direction on the left, and then we sliced it and punched through it in the plane on the right. And we were trying to understand if the behavior is different if you probe it in the fiber direction versus cross the fiber direction. We used meaty. Many of you might have tried it. It's actually a very delicious mushroom root steak, and it has a very great nutritious and ingredient list. So it has only 7 ingredients, as you can see here, and 95% of mycelium. And that's great because it's naturally fibrous in microstructure, and it's also hopefully anisotropic, as we will see in just a bit. It's also very nutritious. It's low in fat, rich in protein, fibers, and vitamins. So it's a very healthy product overall. So now the question is, does it mimic the mechanics of plant and animal meats? And actually, is it anisotropic? So this is our series of tests, where the first row is, again, the shear, the compression test, and the double compression texture profile analysis test. And interestingly, we were very, very disappointed when we first did this. The in-plane and the cross-plane behavior in blue and red lie on top of one another. So that was hugely disappointing, because we expected a very large difference in the two. And we said, okay, it must be anisotropic. There must be some effect of the fiber direction. And you don't see it at all. And we're like, we must be doing something wrong. And so we went back and actually said, well, something is wrong here, because our double compression test takes 800 seconds in this case. So that's 10 minutes, and nobody chews 10 minutes for 2 bites. And so we did increase the rate at the 2 chews. And you can see in the bottom row, we make it faster, 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 faster. And ultimately, the last bottom right picture is 2 seconds, where actually it's a little faster than real chewing. I think 4 is maybe realistic. So that would be chew, release, right? So that's the second to last figure. And if we increase the rate, we actually find a difference. So the faster we probe, the more the difference. And actually, for the very fastest, the blue curves are about 4 times above the red curve. So there is some anisotropy, but only when we test at high dynamics. We did the model discovery with our network, and we discovered one model that's similar to before, the dark brown. You remember that was the Neo-Hope model. That's a linear model. So that's part of what we found. But the best model in just one term is now exponential, and that's called the Demery model. It's a model that people have previously used for soft biomaterials. And it's different in that it's exponential. And you can see that these curves actually increase as you increase the loading. So this exponential feature is captured by this red Demery term. If we allow for 2 terms in the second row, we find a brown and a red term. So actually we find the linear and the exponential term combined. And what's really, really interesting, think about that for a while, is we have these 8 terms that you see here, and that gives you 2 to the power of 8, 256 combinations of terms. And out of these 256, we find exactly the same combination in plane and cross plane. So we find exactly the same model. But we do find different parameters, and that's what you see on the right. So there you see the parameterization of these two models. The parameterization is different because their curves have slightly different features. But it's great to see that actually the trend is pretty similar for both. So here's the stiffnesses in compression and in shear and the mean stiffness. And disappointing for us, they are pretty close in both directions. They are very next to each other, right? The orange is the one across the plane, and then the brown is the cross fiber testing orthogonal to the sheet. And they are all on the order of 30 kilopascals. When you test slow, quasi-statically, it's almost isotropic, and we were disappointed to see that. Then we increased the rate, and this is interesting. So from the top row to the bottom row, we went faster and faster and faster and faster. And if you look at the figures, you almost can see what happens. So if you probe very slow at the top, you squeeze a lot of water out, and eventually you only probe the solid. So the stiffness that you get is the solid stiffness, and that's the 30 kilopascal. Now, if you probe very quick at the very bottom row, there's almost no fluid leaving the solid. So you're probing both the solid and the fluid. And so the response is much stiffer because the fluid is trapped, and you test that. And that's what you see here. So this is the texture profile, the 6 parameters for texture profile analysis at different rates. And to the very left, where it says 2 seconds, it's the fastest test. And where it says 800 seconds, it's the slowest test. And you can see for stiffness, hardness, and resilience, I have indicated through these diagonal arrows, the faster you test from the right of the graph to the left, the harder, the stiffer, and the more resilient the material becomes. And that's because the water that's trapped into the material is also tested, and then gives it more stiffness, more hardness, and more resilience. So that's quite interesting. Comparing this to all our other products, this is nice because now we had these 8 comparison products, and we're hoping that someone comes up and says, hey, I have another product, how does it compare? So we could do the same study if you have any kind of product and place it in this graph wherever it falls and color it in green and say, okay, your product sits in these right here. So if you have anything to test, please reach out. We're happy to do that. So here in summary, what we find is at the very fast pace, the dynamic double compression, we find a huge difference between cross-plane and in-plane stiffness in dark brown and in orange. And it's significant. That's what these little stars on top tell you. And you can see that with the naked eye. And the difference in stiffness and hardness is almost by a factor of 4. So that's huge. Now we said, okay, again, we can taste this, we can eat it, see how we feel when we eat it. So this is the result of our sensory survey. And there, it's actually quite interesting because we went on to do this because we said people think fungi steak would be more meaty and more fibrous. And this is exactly what the survey shows, even with only 16 people. So people perceive it as more chewy. So the brown and orange bars are all to the very left with the highest chewiness factor. They're also more viscous. That's what this test shows. It has this water that's leaving the sample, so it's time dependent. It's also more fibrous and more moist and more meat-like, which is interesting because fibrous by a landslide by more than a factor 2, and then also more moist. And it's actually more meat-like, not only than the plant-based products, but also than 2 animal products, which is interesting. And that's really the umami taste of a mushroom. So in summary, here's the product. Here's all the results. It's in the paper. We'll be submitting it, uploading it in a week or so. You'll have access to that. So this is actually all the findings related to this product. This is the only fungi product we've tested so far. So if you have any other or know of any other, we'd be happy to test it and add it to our database. In summary, fungi-based steak delivers. It offers a nutritious, sustainable, textural, compelling alternative. It has a very lean ingredient list. It's made up of 95% of mycelium. It doesn't need additives or gums or anything to stabilize. It's very nutritious, low in fat, and rich in other components, vitamins, fibers. It's sustainable. It grows very fast in vertical farms at low water use and low environmental impact. And interestingly, what our study shows, it has a meat-like texture because of its filamentous microstructure. It's minimally processed because the fibers are already there. You don't need to extrude to generate fibers. It's naturally meat-like because of the umami taste of mushrooms, and its mechanical signature compares well to plant-based and animal meats. But in addition, it outperforms those products in terms of fibrousness, moistness, and meatiness. So that was actually very cool. All right, so this is our last result, and I'm going to just share a couple of things what we want to do next, just to give you an idea. We're doing all of this because we actually want to use AI for food. And you've already seen our network. And ideally, what we want to do is we want to have a network that maps rheology directly from the test, puts it through the network, discovers stiffnesses and models. That's what we already do. And then map that onto our perception of taste. So if we do that for a large training set, right now we only have our 8 solid and 8 deli meats and the fungi meat. But if we do have more more products here, we can actually have a pipeline and train it on a broader data set and potentially predict the outcome when we just see the rheology. This is the simplest step, and that's probably something we can do within the next couple of months. The North Star of all of this, as many of you know, is to predict consumer preferences from formulations. So if we have formulations, meaning ingredients and weights of the ingredients, can we predict how people perceive the taste, the flavor, and the acceptance of those products? So that's something we can do if we have a lot of data, but then the real holy grail would be to reverse this and say, if we have a request for a certain flavor, can we discover a formulation that matches that flavor? So that would be inverse. And there's any inverse simulation, any inverse problem, it would require huge data. So we're trying to collect more, we're trying to reach out and work with other partners and generate more data sets. So you can actually try to come closer to predicting ingredients and weights from formulation. Fungi might be a good start because it has a very lean ingredient list. If the lists are very long, it might be difficult to do this. So these are the people who helped do the studies. On the left, you see Skylar and Reese and Thibault. So those are the students who led the student studies. And then there's a lot of people who advised, some professors. And then on the right is our analysis team. So those are the people who run things through the neural network. Jeremy, Kevin, Denisa, Hagen, Matthias. So all of them are running the machinery and programming the networks. And then again, below you see the links. If you want to reach out, we're happy to respond to that. One last thing I'd like to share is we have a new initiative at Stanford, and that's called Food at Stanford. It's a campus-wide initiative, and there's a couple of people here that are part of it, and a lot of more that are not in the picture. You might recognize a few individuals. So in the green jacket is Pat Brown. He's a founder of Impossible Foods. He's part of this. He's back at Stanford as a professor. And then at the very bottom right is Christopher Gardner, for example. He's a professor of medicine and the host of the Netflix documentary, You Are What You Eat. You might know him from that show. And there's a bunch of other people. They're all interested in alternative protein in the right sense. There's also Vayu Hilmahini, you might know, because he has given a seminar in this series. He's just not on the photo because he's currently on paternity leave. So you can see him here with his child. All that together, last call is we have a symposium in 2 weeks from now. It's called Rethinking Food from Plate to Planet. We have amazing invited speakers. So it's all external speakers who will share their thoughts on alternative protein and on foods that are healthy for people and for the planet. You can register by next week. It's free, so we support it. There'll be great speakers, and there'll also be great foods, and it's all supported through Food at Stanford. And that's my last slide. Thank you all, and I'm happy to take questions.
Thank you so much for that, Ellen. That was such a comprehensive body of work, and it's so lovely to see how you've connected this testing in your lab with actually working with the students to see how they perceive this and all of that. And thank you for that call at the end to invite people in to the event. It's really lovely to see the initiative that you're building and the connections that you're forming across campus.
Thank you.
We have a bunch of questions in the chat, but there's one that I would love to open with about kind of the complexity of many animal meat forms. So a lot of the work that you started with was kind of these processed meats that you mentioned, and I think they tend to be a little bit more homogeneous. But one of the things that kind of makes animal meat so interesting is the heterogeneity of the texture. And so I'm curious if you could speak a little bit more to that, and also if there are potentially more complex mechanical signatures you might want to look at, or combinations of signatures that could kind of address some of the nuances of animal meat that aren't as represented in processed meat.
This is a great point, and that actually is exactly what we showed with fungi type meats. Those are anisotropic, and we didn't have any comparison so far on the animal meat end, right? So a good thing to do, and probably the next worthy step, would be to test real animal steak and compare that against the fungi steak, and then see whether it matches the fibrous structure, whether the fibers really also contribute the same way. And so that's a limitation. We've compared the only one anisotropic fibrous product that we have in our portfolio to date is the fungi meat. And to compare that against processed animal meats is actually an unfair comparison because, in a way, we need a steak to compare it against. And that's probably the next thing we're going to do. And that's a great point. Another point also is that the nature of these fibers typically comes out most in tension. So we haven't yet tested in tension. So that's another thing we need to do. And you're right. I mean, this is probably the very next thing we'll do over the summer.
Amazing. I really look forward to seeing that. Do you have any predictions for how a steak would perform, and also how that might compare to people's perceived texture, given that the fungal steaks that you mentioned didn't line up quite as well as some of the processed meats?
Yeah. So I, it's interesting because I think it's different. I mean, like you said, there's a huge difference in a sausage or a hot dog, in terms of the rheology. So I we're expecting to see quite some differences, and we think that an animal steak will probably be more closely to a fungi steak. That's what it's trying to mimic. But it'll be interesting to see. The funny thing is that it'll be challenging to find people. We always have challenges to find people who to eat animal meat in the study. So we need to go probably in the dining halls. We find more people that are eating animal meats. Our student group, there's a lot of vegan students, and they don't want to even taste the real animal steak. But you're right. I think that would be an interesting step. And I'm expecting to see very close matches with the fungi steak, really. And also interesting to maybe study more fungi products. We just have had the one.
Yeah, I'll be really curious to see the differences as you test more fungi products. And I think it's kind of a funny problem to have a scarcity of people who are willing to try the meat products. Um, we have a couple questions in the chat that I'd love to start with. So, one is asking for a little bit more details about the rationale for the double compression and chewing twice test. So, is that something that's a field standard, or is that the average number of chews that are needed to break down food?
So that's the traditionally the method by which food scientists characterize foods. It's so we came from a mechanics angle, and we thought it's not very reproducibly characterized because, like you've seen, if you do it at different velocities and compress at different rates, you see very different results. And so what we found is if you go to the literature, the first time it was mentioned was probably in the 50s, 60s, 1950s, 1960s. So it was a very old traditional test, but people only compare data within their own lab. And so they know what they use in terms of all these parameters that you need for the test. And it's very difficult if you read a paper to match what they're doing because they don't always report all the little details. So it's not a standardized test as any other standardized test. And so, for example, it measures a force, and it doesn't always say how big the sample is. If you have a much bigger sample, the force to compress it is much higher, obviously. And so there are a lot of inconsistencies if you go to the literature, but it is the test that people agree to most when you go to the food science community. And it is a test that is commonly accepted amongst food people, I would say.
Awesome. That's really helpful to know. We have another question about designing these experiments. One of our participants shared that it's their understanding that it's common to use samples whose dimensions have a larger diameter than the height for compression, whereas you mentioned having a smaller diameter than the height. So can you talk a little bit about how you chose those dimensions,
Mm-hmm.
and how that impacted the testing that you did?
Yeah, that's actually a great question. So the idea is that the sample should be homogeneous in this test, meaning that it deforms the same wherever you are in the sample, right? The sample is too small, for example, and you need to glue it to the holder. Then there's a lot of glue in the sample, and you test a lot of glue. So you want to make it big enough so that there's actually a lot of sample that's not glued, and the glue doesn't impact your test. If you make it too big, you might have a very local effect somewhere, and it would overshadow the overall behavior. So it's not homogeneous in the sense that it's the same across the entire sample. So it's always tricky to find good sizes. It depends, I think, on the softness of the material. If it's very stiff, you need small samples might work. If it's very soft, it's difficult to get big samples into a machine because they would just fail. So I think it's important to report all the sizes that you're using. We've been using consistently the same sizes in tension and compression and shear, and we report it, and then we extract stiffnesses because the stiffness is the size scaled extract, right? I mean, we're not reporting the force, but it's scaled by area already. So in that sense, it's somewhat normalized. Yeah. But it's a good
Right.
point. You need to somehow consistently do it and report those metrics for sure.
Yeah, yeah, definitely a challenge as you mentioned there's different things that can change with the shape and all of that. So thank you for providing the insight into that. Um, One person asked a question about how these meat analogs were produced, and I'd like to tie that with a broader question, where you started to speak to the role of ingredient lists and how the stiffness currently is often tuned by adding gums and other additives. Have you all been able to start looking at the role of different processing techniques in these mechanical signatures for the both the animal products and the meat analogs?
So I think that's a whole different way to tune stiffnesses, especially in the ultra processed products. Is like how they are minced, how they are glued together, how they're produced, how they're extruded. So I think there's a lot of degrees of freedom. A lot of people use AI actually to tune now, not in the past, but there's a trend for a lot of people to use AI to find good process parameters that can be predictive of the final product stiffness or fibrousness. And so those are, I think, great research questions that people are looking into. We didn't do this yet, but I'm hoping that we can do simple products. For instance, there's 5 different tofus of different stiffnesses where we can correlate the way they're made to their stiffness and their perception. So the simpler the product, the easier it is for us to find these correlations. If you have 20 ingredients, it's very difficult to tease out what exactly it is. And then, like you said, the processing methods also overlay an extra layer of complexity of it, right? So it's a great field. We want to start simple because simply we're not real food scientists and we don't have the resources as big food. But I think that's probably what you need to do if you wanted to look at it holistically. Yeah.
Yeah, I think that would be incredibly powerful to be able to connect, even if we're using the same ingredient and we're extruding it versus other
Yeah.
processing techniques. I think it would be so fascinating to see um, how that changes how people perceive it for sure. I'd like to follow up on a comment that you made about aiming to replace sensory panels, which I think is, as you are well aware, one of the most expensive and time-consuming aspects of this work. Really, you mentioned in your own experience, it's hard to find people who will test this for you, and to get consistency around that. So I think that's an extremely valuable thing to be able to offer. So I'm curious about if there are other tests that you're kind of developing or thinking about that we could use to replace different aspects of sensory panels, and also where you see the limits of that. Like, what do you think we will not be able to predict based on the information that you're collecting?
I think that's a very interesting question. I think the taste, for example, is probably relatively simple to predict because there's only the 5 basic tastes. And I guess you can look at the ingredient list and map that relatively reasonably well onto taste. Flavor, as you know, there's thousands of different flavors that we can sense or attribute to a product, and that's much harder to do. Maybe if you narrow it down to certain features, like we did with the 12, you probably have a little more. then I think that that would be interesting. We look mainly at textural features that are related to mechanics, but of course, there's many other flavor features. I should also mention that we have a sequence from Food System Innovations, and they have this huge study, you're probably aware of the Nectar study, where they have a huge database of testing people responses to different foods. So they have, I think, dozens of products, and we actually work with them towards using those data as well. But they have dozens of products, and then they have the biggest possible survey, and they not only surveyed the 12 things that we did survey, but for massive different features. So I think leveraging those kind of data sets, and that's a public one because that's also philanthropically funded, is great because we're trying to actually also build that in, and things that are publicly shared. I think that's very important. That's probably a good time to also make that pitch that what you do is important because it's all philanthropic, it's shared, it allows everybody to use and to connect and to exchange information. And so I think for those to answer these questions, I think it would be good for smaller businesses to connect to philanthropically funded organizations like yours. Yeah.
It's so great that you're connected to all the work happening with Nectar and everything. There's a lot of really
Yeah.
fantastic things coming out of that. Um, Building on this idea of being able to predict how different textural signatures impact perception and kind of this vision that you've presented of even being able to do that in reverse, which I find really compelling. How are you accounting for potential differences across populations, or are you thinking about how cultural associations might play into how people interact with these samples, how they perceive it, and ultimately what their preferences are?
I think it's a huge risk, especially when people think they can use AI to replace. And I think that comes back to your first question. When you think you can use AI to replace people entirely, because I mean, that's like how predictive is your model and where does it predict into, right? If you can, it's very difficult to predict the behavior, just a different regional group of people. If you test, so for instance, we want to do tests at Stanford in the dining halls. That's a very special subset of people. It's all young people. We've seen they're very open-minded to trying new things. They live in California. They probably don't eat a lot of meat to begin with. And so it selects for people that have a very different preference than probably if you go to different parts of the country or even yet a different country. So I think you're right. You have to be very careful where your data come from and what group of population is associated with the results that you have tested. Yeah.
Yeah, I think that is another reason why the open source technique that you're mentioning and that you're leaning towards and collaborating with others to work on is potentially really powerful because then you can also source data from beyond Stanford's campus and hopefully get a bigger representation of populations and things. But I do think that's a big open question for sure. So it's great
Absolutely. Yeah.
that you're thinking about it. We have another question here, kind of about the boundary conditions when testing, in both in compression and testing. So, what role can frictional forces or tribology play in predicting consumption? And is there room for including, for instance, the role of saliva in these modeling objectives?
We've actually asked ourselves just recently. I talked to someone who said, well, there's a huge impact. Chewing is not just the mechanical breakdown of the sample, but essentially what you're doing is you break it down, you mix it with saliva, it becomes a different material, essentially, from a material science point of view. And then the more you chew it, the more actually the effect of saliva carries through. So I think in tests, you can probably include that in some way. If you have a liquid that has a composition like saliva, you add it to your test. um, in our case, we just wanted to have something relatively simple and reproducible. It's probably not the most realistic. If you want to be more realistic, you give up a lot of the simplicity and reproducibility, but you probably be more authentic to a real tool. That's a great point. And then in terms of boundary conditions, so I think there's room for all kinds of tests, like I've shown. I mean, we've done tension, compression, shear, people have done rheology, but these are not people commonly in the food science community. So those people do the texture profile analysis mainly. And I think it's good for people out of the, what you just said, the rheology, or there's all kinds of other small tests, right? Tribology, indentation, all kinds of levels of size of can indent small pieces, you can indent like just the fibers of the fungi material. So I think all of these people could contribute their knowledge to this to understand it better.
I know there are entire lines of research that are just developing artificial tongues and things like that to start to look into that because it's a complex problem and there's chemical breakdown that starts and all of that. So fully
Yeah.
understand wanting to start with a simpler version of the problem to see if this is even, if it even works.
But I think the cool thing is we're at a point where all these different research directions are somehow um, coming together, and it's so
Mhm.
great to see the community build from all different backgrounds. And I think that will provide a lot more insight rather than just having like everybody siloed in their own research bunch, right? So this is a great
Absolutely.
time.
Absolutely. Kind of leading from that, you mentioned like indentation, texture profile analysis, all of these techniques that are somewhat commonly used in industry. Is there a way that you see your work kind of plugging into the current formulation process, given that um, most companies are going to be working with a pretty limited data set and often sort of have some hesitation around sharing their information? Like, is there a way that you see yourself interfacing with existing uh, industrial formulation efforts?
So we're huge fan of open source. So our tests I know are open source. If anybody wants things tested, I think our condition would be to make it open source. And that seems to work well for smaller, like startup type of level companies, but bigger companies, obviously, like we're also starting to talk to Notco, for example, but they have this decade-long experience. They're amazing, but it's also understandable that that's their asset and they don't want to share on the data. So I think that there has to be some kind of way to collaborate, and we're trying to work out ways to collaborate beyond sharing data. So there's other ways to work together, share technologies, share methods, share experience. And so I think that's the way we try to collaborate with Notco. And there's other companies where we can do things without having to explicitly share data. But then again, if anybody's on this call or watches this and wants to have stuff tested or work, please reach out. We have a lot of resources. We have students, they're super eager to test, and they're there all summer. And they'll be happy to work with you if you have anything to test or work
That's such an
together.
incredible offer. Such an incredible offer. And kind of building on that call to action that you've put forth a couple times, are there kind of types of data or types of products that would really help you build this out further? Like I know we were talking about steaks, for instance. So if you think about making a map of the kind of stiffness profiles that you've been able to test
Mhm.
so far or have represented, what are the edges where you're like, we don't really have anything in this space and we really need either people to bring us their things or, hey, people should start formulating this so we can test it.
Yeah, I think fungi would be great because you only have one. It turned out to be nice to have always four in each category because then you can compare against each other. And then, like you suggested, anything anisotropic, like steak, is probably something we're going to do next. And then probably anything that comes our way, we're happy to test. It doesn't necessarily have to be meat. We just started with meat right now. It could also be other products. But yeah, and I think to have multiple of one type or family is nice because then you get a better sense than just a single one product, obviously. We're working with Nectar, like we said, and then Nectar has all this huge base of products that they have surveyed people on. So that's probably a list that we could go down naturally and find
Mhm.
products that they have already good survey data for. Yeah.
Awesome. Awesome. And if you think about building out this long-term vision that you mentioned of using AI for food and really being able to have this predictive modeling and all of that, how are you thinking about kind of building out all the infrastructure for that? So right now, it's fantastic. You're doing all of this work in your lab and things like that. Do you think that is something that would be sustainable over the course of like building out the data set you would need to really kind of model effectively and even get to that inverse design problem that you mentioned? Or do you have a vision of like, oh, in an ideal universe, it would be a network of hubs in this way? Or how are you thinking about that problem?
That's an interesting question. And it's kind of like asking how far can you go in academia in the setting that you have with the number of people you're limited, with resources you're probably limited. So I've done this for the heart, and that actually gives me kind of the confidence to do this again. So we have created codes to model the heart, like really algorithms. And we've shared them with a finite element company, and they have made it publicly available. They've created a platform that's now being used by hundreds of people all across the world to crowdsource them. And there's even the FDA, the Federal Drug Administration, or other agencies, regulatory agencies, development agencies. Everybody's using this. And at some point, we just discovered, okay, we are too small to do this on our own. We donated the code to the company, and they built it out, and we consulted on them how to do it, and still working with them. But this has grown to about an ecosystem of about 400 users worldwide. And so I think this would be a kind of an exit, not even exit, but a strategy to kind of make this more translatable and translational, right? To make it accessible on a larger scale.
Yeah, it's a really cool vision. And it's also so lovely to hear about your background addressing other problems and how that's informed the design that you're taking on now. Moving from the heart to steaks is, I think, a very fascinating trajectory. That's so cool. Um, yeah, very interesting to think about. We have a question here that I think is pretty fascinating about whether any of the learnings from this research could be transferred to thinking about pet foods or non-human food experiences and sensory experiences.
Yeah, I think that is probably true. I mean, I don't see why it shouldn't. It can probably be translated to either animal foods, but also to other food products, right? It doesn't necessarily have to be meat. So the technology is pretty broadly applicable. Um, a survey on dogs would probably be difficult. Just thinking, what can we not translate? But I think the probing and all that would probably be feasible. Yeah.
Great. Awesome. Thank you for that. I think it would be very amusing to watch the videos of sensory testing with puppies, so I hope that happens one day.
Yeah, that would be a nice project. Really exciting. Yeah.
I'd love to hear a little bit more about the initiative that you're building at Stanford. I know you kind of gave us a high-level overview quickly in that second to last slide, but could you speak a little bit more about what you're trying to build and who you're trying to bring in as you do that?
Yeah, so we just recognize that there are so many people on campus who do food related work broadly. And the cool thing about Stanford is we have all different kinds of schools on campus, not just a technical school or a technology school. So we have engineering, and we have the humanities, and we have medicine, and then we have a DER school, which is a sustainability school. We have education, we have law, we have business, so 7 schools. And I think we need all of that if we want to advance really in this field. It's so important. There's people like Pat Brown that think deeply about the environment, and they really want to eliminate animals out of the food chain entirely. And they can't do that by themselves. They're in a medical school, they need lawmakers and law, they need business, how to translate these ideas. There's education in food is very important, right? To educate the right students or kids how to eat right and healthy. So there's a lot of aspects I think that are very interwoven, and where you can make the biggest impact if you really acknowledge that all these people can work together. And so that's the whole idea. To really span that across the entire campus, and then also at the same time leverage students, because like I said, we're very passionate about our students. We're like passionate about teaching them, but we get a lot in return because they have great and crazy ideas. And so if we absorb that and build on that, I think it could be very, very powerful overall.
Awesome. Awesome. Well, thank you for taking on that really important field building work. I think it's so great to see what's possible in a university setting when you bring all of these disciplines together. And bioex seems like a really unique space to get to do that. And, there's an incredible diversity of faculty, like you mentioned. So it's really great to see the model that you're taking of empowering students, having Skylar's project come out of a class, it sounds like, and then really leveraging faculty to have a far-reaching impact. It's all
But
of our
I should also say, I mean, you've been amazing in supporting student groups, right? And we've talked about this a lot. So just to empower students to work in the space or organize seminars or classes or seminar series in this and support that, I think it's just also very amazing because it empowers them to really make an impact like you've been doing or still doing in many places. So I think that's also a great approach because the students are just so excited about this topic, as you know, from talking to them. And so it's just really driving innovation at a very different level.
Yeah, they are really incredible. And I think your work is a testament to this. And we see this across the ecosystem where students are really driving so much of the amazing work that's happening here. So thank you for that. I think with that, we are rounding out our questions. So I just want to thank you again for taking the time to join us and for that fantastic talk, and for leaving us with little nuggets of invitations for future work to think about. I want to echo the call that Ellen put out. Like, please bring your samples to Stanford so that they can expand this data set and give you some information about what you're looking at. And this symposium is happening later this month, so please check that out. And Ellen, I really want to thank you again for this lovely talk and for joining us today.
Yeah, thank you for inviting me. It's a lot of fun. Great
Awesome.
questions.
Thank
Thank
you.
you.
Thanks, audience. And thank you all for joining us today. I hope we'll see you next month for our next Science of All Protein seminar. Thanks,
Thank
everyone.
you.